Okay, so reaching very near the end. Today we are going to finish up with our understanding of the GI system. So we're going to be doing the last couple pages in that packet on peristalsis and segmentation, and we're going to move right into the reproductive system. Okay. So now that we know a lot, or at least some, about digestion and all the organs involved with our GI tract, we know a lot about how we eat food, how we break it down. And our final topic is just how it actually moves, how that food is going to be moving through your system. So this is where peristalsis and segmentation come in. Okay. So sometimes we'll refer to this as GI motility. So motility is just referring to this movement of the GI tract. And you don't have possibly this picture here. It's from earlier in your slides. I just pulled it up so that I can point out to you some of these things that we're talking about here. Okay, so GI motility is about movements of the wall of the GI tract that are going to allow you to propel food forward, as well as mix around the food that's in your system. Now that we know that our carbohydrates, our proteins, our lipids are all being digested by enzymes, that movement, swirling, swishing stuff together in your gut, is actually really important. We can understand why. We need those enzymes to come into contact with those molecules that they're going to help us break down. Okay, so the GI movement, the GI motility, is primarily due to contractions in our muscle layer. So our muscularis externa was one of the layers in the wall of the GI tract. So this is coming back to the beginning. And we saw that the muscularis externa, this layer, had a layer of circular muscle within it and a layer of longitudinal muscle within it. So when we're talking about how this is happening, we're primarily thinking about those two layers of muscle in different orientations. So we have circular muscle. It's going to act kind of like we've seen sphincters act, right? It can contract, make things narrower, expand, make things wider. And that longitudinal layer, when it contracts, right, it's going to make things shorter. Or if it relaxes, it's going to make things longer. So we have these two ways that we can produce movement of the wall of the GI tract. So we want to be able to mix together all our enzymes, all our juices, create that chyme so that we can have digestion happen. And we also want to propel the contents of the GI tract forward so that it doesn't say stuck in your mouth. We want it to move down the esophagus, through the stomach, through the small intestine, through the large intestine, and out of your body. We don't want it just piling up. Okay. So, looks funny now. All right. So when we're talking about peristalsis and segmentation, we're splitting up essentially kind of these two functions. So peristalsis is going to refer to that propelling the food forward. So moving it forward, moving it from the small intestine to the large intestine, out the body. That's peristalsis. So if you've seen this word, I think the first time I saw this when I was a kid was when I was thinking about how a snake eats, right? That's the peristalsis, the ball of food moving down towards the end. Okay. So this is going to be waves of contraction in the muscularis mucosae in response to basic electrical rhythms. So this is like, think about like our pacemaker cells, right? Some of the smooth muscle has a similar property where we are forming these potentials essentially. And that's where these contractions are coming from. We're not going to go into any further detail on that, basically. But you don't have to think about it. You don't have to contract your small intestine. It's just going to happen. Segmentation is how we are going to be mixing together the contents of our GI tract. So we're mixing together food and all these juices to make the chyme, essentially. All right. So in peristalsis, we're just trying to move the ball of food forward. The way this is going to happen is we're going to have an interaction between what's happening in the proximal segment. So the segment near the beginning, so say up here, and a distal segment here. So we can think about sort of chunks here, although we can see that it's actually kind of more of a smooth gradient going on. Okay. So all we're doing to move food forward is in the proximal segment, we're going to narrow our GI tract, narrow this tube. And as we push in from the sides like that, this just moves that ball of food forward, right? So you're squeezing behind it. Think of it as a marble, or it's just going to roll forward, okay? So we're decreasing the diameter of this proximal segment, and we got to make space for that marble to roll forward. So in the distal segment, so the second chunk, we're going to have some relaxation going on. So this circular muscle is going to be relaxing, making this wider so that we can have nice, smooth movement, right? We don't want to produce a lot of resistance to this movement. We want to let it happen easily. So that's the circular muscle relaxing. The other thing that's happening is the longitudinal muscle is contracting a bit. So think about it like, I don't know if you've ever had to put a hoodie string or something back in, right? You kind of crunch. If you go home and like play with a hoodie or a drawstring to try this, right? You kind of crunch the tube to get the little pokey end to come out. That's why the longitudinal muscle is contracting, right? You're kind of trying to crunch it down to move this ball of food even farther forward. Okay. So this is really all we have to do in order to propel food forward by peristalsis. We are squeezing behind our ball of food, pushing it forward, and also crunching up that forward part, that distal part a little bit so that we can move it even farther. So that's our peristalsis. And then in our segmentation, we're mainly thinking about this in the small intestine, actually. And I say actually because when you've seen the intestines in lab, right, actually it looks like the colon is more segmented when we look at those haustra. But when we're thinking about the physiology of it, segmentation is really important in the small intestine because that's where the digestive processes are happening, right? That's where we're really concerned with all the enzymes and like our pancreatic juice coming into contact with all the macromolecules in our food. Okay. So this is primarily going to have to do with the circular muscle layer. And basically what we're doing here is instead of moving the ball of food just forward, we're kind of jiggling back and forth a little bit. Okay, so we're going to be alternating contractions between intestinal segments. So if we imagine one chunk of food here, one chunk of food there, we're going to have a lot of circular muscle contractions, kind of pushing both back and forwards until we get mixing between those two compartments. And that's kind of all that we need to understand conceptually about this, so that we're blending all this stuff together. All right. We know that the chyme already coming from the stomach is a blend of those gastric juices and our food that we've chewed up. That chyme is moving forward into the duodenum. In the duodenum, we have our duct bringing forward our pancreatic juices with all those enzymes for additional protein digestion, lipid digestion. We have another amylase for starch digestion. So even though the chyme is already kind of smoothie-like and liquidy, we also need to keep mixing it so that once we get those juices in, they're not like all just kind of stuck in a clump on the side. Okay. So that's the concept of segmentation. And that's really it for the end of our digestive system. That's kind of the level of understanding we want. So GI motility, important for mixing enzymes, getting food out of your body. If your intestines stopped moving, you'd like stop pooping, stop digesting food, you'd have trouble getting the contents of that food broken apart so that you could take it into your cells. So we're going to move directly into the reproductive system. So we got a part 1 and a part 2. This is where we're going to hang out for the rest of the year. Okay. So we're going to start with an overview of reproductive physiology. And in particular, we're going to begin with kind of developmentally how we end up with different structures in the reproductive system. So we're going to begin by thinking a little bit about gametes, and we're going to be thinking about sexual reproduction, so fertilization. We're going to think a little bit about genes and gene sorting and packaging. We're not going to go too deep into genetics. We're going to be mainly focusing on how that influences hormones that move us forward towards one sort of plan or another. We're going to walk through the components of the reproductive system. So you either have already or about to see these in lab. Is the final lab unit done yet? Not in yet. Okay, great. So you've seen the components of the reproductive system. We'll go over them again. And then we'll talk about events following fertilization and changes in reproductive activity over the course of the human lifespan. I just want to make one note before we go into this, because I always worry a little bit or get a little bit tripped up as I'm trying to use precise language with you, but also be understandable. So I just want to make a note that as we talk about male and female anatomy and physiology in this PowerPoint, there's a lot of other stuff that goes into people's lived experiences, gender identity, gender expression. Also, many of these things are spectra, and that gets really complicated, and we have 4 days till the end of the school year. So we are not going to be going into all the cool variation that there is, but it exists. We're just going to be doing very basic, typical kind of foundational stuff that you see in most people at birth. So wherever you are in this, if I say something that upsets you or that you find problematic, let me know and I'll try to change my language. But I just want you to know that that's where we're kind of starting from. And as a personal note for like all of you going into healthcare fields, my takeaway from all of this is, right, life is complicated. Everybody's going through their own stuff. A&P is complicated, really complicated, as we all know now after going through this course. Be nice to people. People are just trying to live their lives. That's it. Treat everybody with dignity and respect. Not that hard, even if we don't understand all the little bits that go into it right away. Okay, so that's my little spiel. I'm not real comfortable with pronouncing to you guys in that way, but thought it was important to say. Okay. So we are going to start by thinking about sex chromosomes and sex hormones and how that affects development of sexual characteristics. Okay. So we're going to start by thinking about reproductive system. We're thinking about how we develop different reproductive organs. So when we're talking about reproductive organs, we're thinking about a wide array of things. So we're thinking about organs involved in mating, in sex, in copulation. We're thinking about organs involved in gametogenesis, so the production of sperm and egg cells, and other functions involved in producing offspring and feeding those offspring. So when we use the word gonads, the gonads, we're going to be talking primarily about the structures that are involved in gametogenesis and secreting sex hormones. So in male anatomy, those are the testes, and in female anatomy, those are the ovaries. There are also accessory reproductive organs, and we'll start mentioning them as necessary as they kind of develop and become part of this story. Okay. So hormones are really important when we're talking about the reproductive system. Now, the testes just produce one type of hormone, androgens. So that root actually, andro, I think is Greek. That always means like man. Okay. So the testes produce androgens, but I will note that androgens are actually found in both males and females because they're also produced by the adrenal cortex. Okay. So it's not true that men only have one type of hormone and women only have one type of hormone. Okay, there's some interplay here. The ovaries produce two types of hormones. They're going to produce estrogens and progesterone, and we're going to be looking in detail at when they produce each of those and how this kind of cycles. Okay, so that'll be in basically the second half of this chunk of material. Okay. So on the whole, males have more androgens and females have more estrogens and progesterone. But there's a mix, and all of these steroids, they're all steroid hormones. So they're all kind of like based on cholesterol. We'd see those rings linked together with little side groups. Okay. You'd find them in both sexes in different proportions. Okay. Accessory reproductive organs are going to be things like organs of the reproductive tract. So we are also thinking about some of those as we're thinking about development. So we're going to be thinking about these structures that are going to transport gametes. So structures that will move the sperm, structures that will move an egg cell. Okay. We also have glands that secrete fluids into the reproductive tract that are important for nourishing some of these structures. So we're going to sort of begin with fertilization and pregnancy, and then we're going to loop back to it at the end again. So right now we're talking about it because we're thinking about, okay, so how do we build a body plan? Okay, how do we develop that? All right. So copulation is a technical term for the act of mating. So for sex, penetrative sex, where we have a sperm deposited into a female and an ovum that may be fertilized. Okay. So after fertilization, pregnancy is going to occur, or gestation is a technical term for that. In the first 2 months, we refer to the developing structures as an embryo. After 2 months, we begin to refer to this as a fetus. There are actually many more terms if you were taking an embryology course for specific phases in this. At the very beginning, we actually have specific words for, okay, is it 2 cells? Is it 4 cells? Is it 8 cells? So this is the difference between embryo and fetus, but it's not that simple as you move forward. After about 9 months, we reach birth, otherwise known as parturition. That's the technical term there. Okay. So we're beginning with fertilization. And right now we're concerned with how do we develop different structures. So this is going to begin with chromosomes. So hopefully in some of your previous classes, you mentioned that humans have chromosomes. We have our 2 sets, and they're divided into autosomes, which are the chromosomes that all of us have. They're the same. You have 2 pairs. And we have our sex chromosomes, which differ. So you can have an X and an X and a Y, or in special cases, different variations upon that. Yeah. So when we are thinking about development of anatomical and physiological structures, biology kind of thinks about like, is there a Y chromosome or is there no Y chromosome? This is because on the Y chromosome, you find something called the SRY gene. So the SRY gene is on the Y chromosome. The Y chromosome is is always smaller, has fewer genes than the X chromosome, which actually, I won't get into too many details, but this can cause men to be more predisposed to certain diseases because they don't have a second copy of a lot of genes. But the SRY gene is one of the important ones on this little chromosome. Yeah. So it's called the SRY gene because it is the sex determining region of the Y chromosome. Very creative naming convention here. Okay. So SRY. So if SRY is present, this is going to set you down the road towards producing testes. And if the SRY gene is absent, you're going to go ahead and produce ovaries. Kind of doesn't matter what else is going on with your chromosomes. If we don't have this gene, ovaries. So actually the very baseline plan for human development is to produce ovaries. Okay. However, when the SRY gene is present, what it codes for is something called testis determining factor, right? So it is a factor that is determining whether you have testes or not, okay? Again, very, very creative naming here, right? So if the SRY gene is present, when your DNA gets read, codes for this testis determining factor. And then once you have testis determining factor running around in your system, this is going to go ahead and cause a fetus to develop testes. If we don't have testis determining factor, then ovaries are going to develop. So you can start to see how some variation is possible already, right? You might just not have a Y chromosome and therefore not have an SRY gene, or you might have a faulty SRY gene and therefore not produce testis determining factor, even though you do have a Y chromosome. Or as we move forward, you might have an SRY gene that works, produces testis determining factor, but for some reason, it's not getting to the cells it needs to. This is where some of our variation in biology comes from. Okay. So sex differentiation is controlled by hormones. And it starts with these beginning signals from the genes. Okay, so we have that SRY gene, testis determining factor, determining whether you're producing testes or not. And then those testes are where a lot of androgens come from and move us forward. Okay, so we're going to get a little specific about embryology right now. So in the first 2 weeks, we consider an embryo to be sexually indifferent. So it doesn't care, it's not male or female, it just has the precursors to both tracks. Right? So we know, right? We have ducts called Wolffian ducts, and these are precursors to the male tract. And we have Müllerian ducts, and these are precursors to the female tract. And by tract, I mean like a tract that's going to develop these testes, these ovaries, as well as all the tubes connected to them that allow you to actually move sperm out of the body, move the ova around after ovulation, things like that. Okay, so we have both ducts. The embryo is sexually indifferent when we start out. It's just waiting for that signal. It's just waiting to see, is that testis determining factor there because of the SRY gene, or is it not? So then we get to sex differentiation. So after this period, after these first 2 weeks, that SRY gene maybe starts getting read, you maybe start determining, or you start producing this testis determining factor. Okay? If you produce this testis determining factor, you're producing testes, and then those testes are going to begin secreting hormones. Okay, so they're secreting hormones. And the testes specifically produce testosterone, right? So test for testes, arone, kind of tells us it's a steroid. That's a typical ending for steroids. So testosterone stimulates the development of those Wolffian ducts. So if you have those developing testes, they start producing testosterone, they're going to tell you, okay, let's actually grow the Wolffian ducts into eventually adult anatomy. And they are also going to be secreting something called Müllerian inhibiting hormone. We're going to abbreviate that MIH. Okay. So Müllerian inhibiting hormone is inhibiting the Müllerian ducts, right? So in addition to saying, check mark, yes, work on developing those Wolffian ducts, we're also sending a signal that says, and actually, we don't need those Müllerian ducts. Let's kill them. Let's have them die away. Okay. So you're both saying yes to the Wolffian ducts as well as inhibiting the Müllerian ducts. Okay. If those hormones are absent, right? So if we don't have functional testes in this developing embryo, we're not going to have hormones, which means that the body is just going to continue with its natural baseline plan. The natural baseline plan is that the Wolffian duct regresses. So naturally, if you don't get testosterone in order to stimulate the development of those ducts, they're going to start to die away. Okay. And then the Müllerian ducts are going to just keep going. Okay. So this is how we would end up with a base plan for female anatomy, right? It's just, we didn't see testes, so we went with what we had. So here's kind of the structure of those ducts. Okay. So we're beginning here in our sexually indifferent stage. So we can see that we have the Wolffian duct here, right, with a primitive gonad attached to it there in yellow. Right, and that duct going down to an opening. And we also have the Müllerian duct to the side of it, right? Also going down to an opening, right? So we're ready to build either body plan, okay? Then we're waiting for our signal, and our signal is development of testes or not, and the production of those hormones. So if we have testes, they are going to produce testosterone and MIH. So testosterone telling us to develop the Wolffian duct. So we can see here that that Wolffian duct is staying. And it's starting to look a little different. So it's merged with this gonad, right? And we can see, actually, I should have kept the colors consistent. Sorry, y'all. Okay. So there's our Wolffian duct continuing on. And because we had that MIH present, that Müllerian inhibiting hormone, we've started to create degeneration of the Müllerian duct, right? So that dotted line tells you this is going away now, right? We're sending it a signal to say, we don't need this, okay? And this is what eventually produces our adult anatomy. So you can see some growing glands, right? So eventually those testes will descend down and out of the body actually, right? But they're going to remain connected to a set of tubes, which we'll call our, sorry, I keep not keeping the colors consistent. Okay, so our testes are still connected to a set of tubes, which is the adult form of our Wolffian duct, taking us out to that opening, right? So that is the resulting structure when we have testes present and when we produce testosterone and Müllerian inhibiting hormone. Okay. On the other side, if we don't produce those hormones, we just go with our Müllerian ducts. So we can see the Müllerian duct from here, right, this one, right, is now beginning to develop some more female anatomy, right? So it's now producing a uterine tube and creating a swelling at the base there, creating a uterus, right? So this is what we have at about 8 to 9 weeks, right? And then eventually we get to the anatomy at birth. So you can see that those tubes kind of fold down and around and come to hug our ovary. So ovary, and we got those tubes still. Our fallopian tubes, our uterine tubes, going to the uterus, down and out the body. Okay. So that's how we develop one or the other of these body plans. Now, there are lots of changes in anatomy and physiology over the course of life, okay? So it's not just development in utero, right? You're not done at birth. We all know that, right? We've all been out in the world for a couple of years. So some really important phases of these changes are really noticeable. So the first one is going to be puberty. So puberty usually starts between 10 and 14 years, and this represents sexual maturation. So even though we have the basic organ systems present at birth, they're not functioning as they do in an adult, even though the anatomy is there. So we need time for the reproductive organs to mature, okay? So, and when we say mature, we mean they have to get to the point where they can produce gametes, so functional sperm and fully developed egg cells, although we will see that the original cells for those eggs are all present in females at birth. Okay? And this is the point at which secondary sexual characteristics will start to develop. So those are the more visible kind of accessory sexual characteristics of the body. So not the reproductive tract itself, but things like vocal changes, changes in breast tissue, changes in hair growth, things like that. So during sexual maturation, so after puberty, after this period where we start to see these changes to the anatomy and physiology, the female reproductive system is going to begin cycling. So we'll talk in detail about the steps of the menstrual cycle, but that's what's happening, okay? Males are now able to produce functional sperm, okay? And males continue with that ability to produce functional sperm over the course of their lifetime. But females are going to undergo a second really important change in their reproductive activity, which is menopause. So during menopause, we have a loss of female reproductive capacity. So we're going to see a loss of that cycling of the female reproductive system. So we'll talk about that later on. So now we are going to move forward, and our first topic, we're going to start with spermatogenesis and some more hormone stuff in adult male anatomy. So we're going to take a look at some of the structures that you have hopefully seen in lab, but we'll do a little review. Okay. So when we are talking about reproductive anatomy and the gonads, the most important piece for our purposes is the testis. So this is the male gonad located within the scrotum and outside the pelvis, outside the rest of the body. This is important because we'll see that the development of sperm actually has a slightly lower required temperature. And that's why we have to have this hangout outside of the body. The testes also are producing androgens, right? So around the testes, we have the tubes that are the adult form of that Wolffian duct. So we can see the epididymis sort of sitting like a little kind of seahorse on top of the testes and turning around with its tail, leading us into the vas deferens that we would see in our spermatic cord, right? We saw that going through the inguinal canal, right? That vas deferens is looping around. We're going to see posterior to the bladder is going to come together with a duct coming from the seminal vesicle, which is a gland, okay? It's joining together with that. We're going to see the ejaculatory ducts now where they've connected. This is all dumping in to the urethra as it travels through the prostate, okay? And then they've joined forces. That urethra continues out through the penis, okay? And we have some more contributions as it goes through, particularly from our bulbourethral gland. So we have another gland putting a little bit more of our fluids into that urethra here. So this is the course that semen will take, sperm will take, and pick up these other things that we're going to talk about what's coming out of those glands, right? By contrast, right, we had our bladder here, right? So we know that urine is also traveling part of this pathway. So coming through the bladder, traveling through the prostate, right? And then it's sharing the urethra essentially with our tract for sperm and other fluids, okay? So this is a little different than what we will see when we look at female anatomy. There's no like sharing of the urethra there. Okay, so we'll contrast when we get to that. All right, so those are some of our reproductive organs. All right, so here we have a zoomed in view of a testis. Okay, so the testis is the male gonad, right? So we can see those gross anatomical features. By gross, I mean big, not like gross, like in normal English. Okay, so we can see that testis there, right? And we can see the epididymis sitting on top of it. looping around. Okay. And now we can see some internal contents. Okay. So within the testis, we actually have 250 to 300 little compartments. Right. So we have tiny little compartments, tiny little sections all through the testis. Okay. So those little compartments contain the seminiferous tubules. So the seminiferous tubules is this little ramen looking stuff here. So these are tiny little tubes where we are going to see the production of sperm, right? So we can see those seminiferous tubules in compartments in the testis, and we can see how they're connected to the epididymis and then connected to the rest of this duct, essentially. We are going to look at a couple cell types here. So we've looked in and cut across a seminiferous tubule here. So think like we took a slice like that. Okay. So now we're looking at the cells lining that seminiferous tubule. So the seminiferous tubule has a lumen. So remember, a lumen is like a hollow part. Okay. So the sperm are going to be eventually in the lumen in the center or hollow part of the tube. The tube is going to be surrounded by Sertoli cells. Okay. So here we see Sertoli cells. We can also see some more cells kind of around this structure called our Leydig cells. So those are over here kind of outside and around, right? Those are the purpley blue kind of ones. Okay. So all of this is inside a testis. Okay. So the Leydig cells, we might think of as interstitial because they're kind of between the tubules, right? They're between tissue, interstitial, but they are cells. They are their own cells. They're secreting testosterone. So we already know that testosterone was important for the development of this reproductive tract in the first place. Okay. They're going to continue secreting testosterone. And those Sertoli cells surrounding the lumen, forming kind of that wall of the seminiferous tubule, you can think of as like modified epithelial cells. So they're forming a seal around the tubule. And they're also supporting the development of the sperm. Okay. And if we zoomed in even more, right, so zooming into this compartment, we're going to be tracking what happens here kind of at the top, at the outside, and how we actually end up with the sperm inside the tubule. So we'll go through that. And here you see some histology. Stuff is pointed out. If you love histology, go through it. I'm not a big histologist myself. Okay. So also around these testes, we have smooth muscle. And this is important because the sperm have to travel. They actually have to travel kind of a long way, right? So we are going to see this function of peristalsis in the testes and the seminiferous tubules as well, and continuing kind of throughout this reproductive tract, right? So just like we just talked about peristalsis, contracting, propelling stuff forward in the GI system, same thing is going to be happening to the sperm, at least at the beginning. Okay. So there's a layer of smooth muscle here so that we can have that happen, right? So we got that smooth muscle there. Okay. We sometimes talk about something called the blood-testis barrier. And all that really is that we do have tight junctions here. So tight junctions create like a good seal. These ones are actually kind of tight, right? So we're kind of protecting the developing sperm and these sperm that we're storing from other things that might be in the body. So we're going to have a luminal compartment and a basal compartment. They're going to be separated by tight junctions. Right. So thinking more about those cells. Okay. So the Sertoli cells specifically, those ones that were lining the tubule. Okay. Their job is to support sperm development. So what they're going to be doing when we say support sperm development, right? They're not just a cheering section. Okay. They're going to be secreting some fluid. So they're secreting a fluid called luminal fluid. So go into the lumen, right? We got watery stuff in there, luminal fluid. They're also going to be secreting something called androgen binding protein. Okay. So androgen binding protein is going to help us keep a steady level of androgens in the lumen. Okay, so we want to be able to keep our hormone levels essentially consistent around the sperm that we've developed. Okay. Those Sertoli cells are our targets for testosterone, which we saw being produced by the Leydig cells, those purpley blue ones, those interstitial cells, essentially, as well as something called FSH, or follicle stimulating hormone. And we'll talk about the pathways with that in a little bit. That's also going to be an important part of the female reproductive cycling. Okay, so this is going to be more of an endocrine signal coming from elsewhere in the body, okay? Or at least involved with a lot of other places in the body. So these target cells, these Sertoli cells, are the target cells, and they're going to secrete chemicals that are going to help us stimulate spermatogenesis, which is the development of the adult sperm, the mature sperm. Another thing that they're going to be secreting is something called inhibin. And this is going to let us do a negative feedback loop on FSH. So FSH is a hormone. We are regulating things here. Inhibin is going to allow us to kind of suppress that signal as well when we need to. I kind of ran through the external genitalia on that picture a little bit, but the external genitalia, our male reproductive organ, is the penis. So the male copulatory organ, you're writing it in a professional note. Its official job is sperm deposition, so depositing sperm for possible fertilization. At rest, the penis is flaccid, so floppy. During sexual arousal, it undergoes erection. So anytime we've seen a muscle with that rect, right, root in it, I guess this is kind of late to tell you if you didn't notice this before, rect, we're thinking rectangles, we're thinking straight lines, right? So rect, when the penis undergoes erection, becomes a straight line, sticks out. Okay, and this is because blood flow to the penis increases and blood actually gets trapped within the penis, right? So it's this swelling of blood, this increase in pressure, essentially, that is causing that change. Okay. We had our ejaculatory ducts where we had our vas deferens meeting up with our seminal vesicles. We looked at how that connects with the urethra within the prostate. Okay, so during ejaculation, the sperm gets ejected through the urethra, so through that tract that we traced through. Also part of the external genitalia, we have the scrotum, so hanging outside the body, suspended beneath the penis. This is where we house testes. This is because sperm can't develop at body temperature. So at 98.6 temperature, too hot for functional sperm. So this allows a cooler environment for sperm development, the fact that we can hang those testes outside the body in the scrotum. And actually, we have musculature within the scrotum and these associated structure in order to kind of change the height and level at which those testes hang in order to thermoregulate. So in order to get just the right temperature. Right? So if we're thinking about the tract through which our sperm are going to travel, they're being produced in the testes, in those seminiferous tubules. They're going to end up in the lumen of the seminiferous tubules, so in the hollow part inside the tube. They're then going to go into the rete testes, so just kind of an extension part of those tubes, into the efferent ductules, which then eject the sperm into the epididymis. They travel through the epididymis into the vas deferens, eventually continue into the urethra and out of the body. So thinking about those structures, we do have accessory glands associated. Okay? So the accessory glands that we're interested in typical male anatomy are the seminal vesicles, the prostate gland itself, and the bulbourethral glands. Right? So the seminal vesicles, their job is to secrete an alkaline fluid. That alkaline fluid has within it some important things, some important components. So it's going to have fructose, so a sugar, right? That can actually feed the sperm and help them get energy in order to do their swim, swim, swim action. We're going to have some enzymes in there. We're also going to have some hormones called prostaglandins within that fluid. That's the first gland you would hit, right? Going into the ejaculatory duct. And then that's meeting up with the vas deferens in the prostate gland. That prostate gland itself is secreting more enzymes and some citrate. Okay. We'll talk about what those do as we move forward. And then we continue. We're in the urethra. We're going to have another little tract coming from the bulbourethral glands, which is going to secrete a viscous fluid. So I don't know why we specify that it's viscous. The important thing to note about those bulbourethral glands and the fluid they're secreting is that it's got a lot of mucus in it, essentially. So that's kind of like a lubrication kind of function. So here we have a table of those different functions. Okay, so we'll actually start off here with sperm and their development on Friday.